So MPS Nova is a twinning project led by ITQB and the Nova Medical School, both part of Nova University of Lisbon. MPS aims to establish a European hub of excellence in microphysiological systems, fueled by pluripotent stem cells technology to strengthen UNL University and innovation capacity in the areas of chronic diseases and host microbiome interaction. So MPS is designed to promote knowledge transfer among partners to leverage Nova University full potential to translate basic research in chronic and infection disease and symbiotic host microbiome relationships into clinical applications. MPS Nova-driven partnership of international leading institutions will enhance the exhibiting multidisciplinarity knowledge and expertise of Nova MPS hubs, advance our best practices in research and training, and expand collaborative networks. And if you want to know more about MPS, you can follow us in the social media as LinkedIn and X. And also you can visit our webpage and know more about the projects, the consortium, the staff exchange and workshops, exploratory projects, virtual seminars, as well as all the news about MPS and events. And without further ado, we'll pass the word to Bruno Pereira, which is our first speaker. And Bruno Pereira is an assistant researcher and project leader in the differentiation in cancer group at I3S University of Porto. He did a PhD in biomedicine in 2013 from the Faculty of Medicine, University of Porto. He was awarded an EMBO fellowship at the Institute for Advanced Bioscience in 2014 and a national postdoc fellowship in 2016 and a SEC junior research contract in 2019, starting his current SEC assistant research position in 2023. He has been interested in the role of post-transcriptional gene regulation mediated by RNA-binding proteins in intestinal homeostasis and colorectal cancer, using mice and patient-derived organoids as study models. He has 16 publications, 5 of them of corresponding author, and has coordinated 4 projects, securing competitive funding from several agencies. He is also the president of the Associação Nacional de Investigadores em Ciência e Tecnologia, and today he will talk about colorectal cancer-derived organoids from basic to clinical applications. Thank you for having accepted our invitation, and the stage is yours.
Thank you for the kind introduction. So let me just share my screen. Can you see it?
Yes.
Okay, great. So good afternoon, everyone. Let me start by thanking the MPS Nova project management team for the kind invitation to participate in this virtual seminar series. So also as an external advisory board member of this twinning project, I've been following your activities and they've been quite exciting. So it's really a pleasure for me to be here. So I'll talk to you a little bit about the work we are developing in the lab, and in the context of this training network, I decided to focus my presentation more on the models we are using, specifically colorectal cancer-derived organoids. And I changed a little bit the title, so I'm going from basic to translational applications, not really clinical applications, because this is more accurate. So I hope this does not ruin your expectations. But the idea will be to share with you the models that we are using, how we interrogate them. So hopefully you can also take some insightful tips. And if you think you might find some advantage in using these models to address some of your research questions, please do feel free to contact us, because we are completely open for collaborations. So, as I said, I will be talking about these advanced 3D cell culture models, but we are using them in a specific research context. And we are very much focused on trying to understand the role of RNA-level regulation in gene expression associated with colorectal cancer development. And in this broad theme, we are particularly interested in a class of proteins called RNA-binding proteins that I will call RBPs for short. These are mainly the main effectors of RNA turnover and protein synthesis, and we know there are extensive literature showing that their activity is altered in different cancer types. So today I will try to tell you two stories. One is related with the functional effect of manipulating a stem cell-related RBP for colorectal cancer cell growth and differentiation. This work is very much almost complete, so we are finalizing some experiments proposed by the reviewers. So it's at its end stage. And I will share a second story, which is very much ongoing, more exploratory, where we are trying to understand the potential of these RBPs for cancer therapy resistance. So as I said, we are using mainly organoids, but also animal models and patient-derived tissues, as you will see. Let me just put here the laser. So for you to get a good understanding of the biological context we are working in, I need to show you a little bit about the biology behind the intestinal epithelium. So basically, if I can put this working, sorry. Yes. Our intestinal mucosa has these finger-like projections into the lumen, which are called villi, where nutrient absorption takes place. And these villi are fed by these submucosal invaginations called crypts that you see here. And at the base of these crypts, we find a population of intestinal stem cells. If we label these stem cells and follow them throughout time, we will see that they divide more or less frequently, every 24 hours, and they give rise to a population of actively proliferating cells that divide every 6 to 12 hours. And as soon as they reach the frontier of the crypt, the end of the crypt, they start to differentiate and give rise to all the mature intestinal cell lineages that we find in the intestine, like the nutrient-absorbing enterocytes or the mucus-producing goblet cells. And these mature cells keep being pushed upwards in this kind of conveyor belt-like movement because new cells are always being generated at the bottom. And as soon as they reach the tip of the villi, they will enter into a process of cell death, which is called anoikis. So they are basically expelled out of the tissue and they die. And this process occurs continually throughout our life due to the presence of these intestinal stem cells, which actually makes our intestinal epithelium one of the fastest self-renewing tissues in our bodies. For many years, we didn't really know how to identify this stem cell population or how to isolate it. And this changed in 2009, actually in 2007, when the group of Hans Clevers in the Netherlands identified the very first intestinal stem cell marker called LGR5. And just 2 years later, in 2009, they uncovered a way of maintaining these LGR5 positive stem cells in culture almost indefinitely, thus giving rise to the whole field of organoids as we know it today. So what they found in this seminal work was that they basically needed to comply with two criteria to keep the stem cells happy outside the body. One was to provide a cocktail with specific growth factors in the culture media that essentially activate developmental signaling pathways that are important to maintain stemness, and provide an extracellular support in the form of a matrix. The one that is mostly used is Matrigel, but there are others. And what they saw is that by providing these two conditions, either isolated single stem cells or crypt fragments were able, in a period of 1 to 2 weeks, to give rise to this complex structure that you see in this video that we now know as an intestinal organoid. So this is basically a multicellular structure which replicates the functional subunit of our intestinal epithelium. So you see these protrusions here around the organoid, which are in contact with the extracellular matrix, which are equivalent to the intestinal crypt, where we can find stem cells and proliferating cells, and they all feed into a common differentiated compartment that is equivalent to the villi, where we find the differentiated cells. And these eventually mature, they die, and they are shed into an enclosed lumen of the organoid that has this characteristic dark color that you see here. And the most interesting aspect is that you can dissociate this structure either mechanically or enzymatically, and each one of these protrusions can give rise to a whole new intestinal organoid and thus perpetuate the culture in time. So this was in 2009, and little over 15 years, we can now generate organoids from almost every tissue in our bodies, either using these resident stem cell populations or using induced pluripotent stem cells for specific organs. And what they have in common is that these are self-organizing stem cell-based structures that phenocopy key aspects of cell type composition, but also the functionality of a given tissue. Unfortunately, these LGR positive stem cells are also relevant during carcinogenesis. So a lot of studies have been showing that these LGR5 positive stem cells can be the cell of origin of intestinal tumors, and they give rise, this intestinal stem cell signature gives rise to more aggressive tumors and higher risk for disease relapse. And so soon after the organoid field started to be implemented, we also realized that we could use the same type of culture conditions to maintain these cancer-derived organoids or tumoroids in this same type of culture, just tweaking a little bit the growth conditions, the nutrients that we use in the culture media. So why is this relevant? Because colorectal cancer, despite being largely preventable through screening methodologies, especially if we can identify it in early stages, it is still a serious health burden worldwide. So it is the third most incident type of cancer and the second most lethal one. And the 5-year survival rate pretty much depends on the stage. So for localized disease, we have a relatively high survival rate of 90%, but this drops dramatically to 15% in case of advanced metastatic disease. Obviously, the treatment that accompany the patients are related with this staging, and it can go from surgery alone to adjuvant chemotherapy or using more recently targeted therapies and even immunotherapy. So in this context, we have been studying one particular RBP called MAX3A that we previously identified as being linked somehow with stemness features in cell lines. And so we decided to establish the first knockout mouse model for this protein. And we could see that, interestingly, these knockout mice, they have a postnatal lethality phenotype, so they cannot survive past 2 or 3 weeks. And this is because they have serious complications in their intestinal epithelium. And a striking change that we found was that the absence of MAX3A leads to the almost complete loss of this LGR5 positive population. And this affects, obviously, intestinal homeostasis and leads to cell death of the mouse due to malnutrition. But at this point, we question ourselves, if this is relevant for normal intestinal homeostasis, does it have a functional impact in terms of the carcinogenic process? And so this is the first story that I want to share with you. So we started by looking at the expression profile of these markers in animal models. So just for you to have an idea, this is the normal expression profile for differentiation and proliferative markers in the mouse colon. So in the colon, we don't have villi, we have only crypts, but the top 2/3 of the crypt is a differentiated compartment, as you see in this scheme on the right. And we can look at the expression profile of different markers and see this segregation in terms of spatial localization. So we have Alshenbuhl positive goblet cells, which are essentially located in the top 2/3 of the crypt, or even PPAR gamma, which is a differentiation marker that we have also identified before as being possibly related with MAX3A. And then you have proliferative marker like EI67, which is basically located at the bottom of the crypts. And then the LGR5 stem cell population, really restricted, and also the MAX3A positive population, also restricted to the stem cells located at the bottom. So this profile changes dramatically when we look at a classic colorectal cancer mouse model, the APC-KRAS mouse model. So here we have two driver events, the two most common driver events for the development of colorectal cancer, which are inactivation of the tumor suppressor APC and activation of the KRAS oncogene under the influence of a query recombinase that is being controlled by an intestinal specific promoter. So basically we can generate colon carcinomas in these mice in a quite fast time, so around 9-10 weeks of age. We see that these adenocarcinomas have overactivation of the Wnt pathway, here indicated by expression of beta-catenin. They also have a lot of proliferating cells, and they lose the differentiated Alshenbuhl positive goblet cells. I think you can appreciate the striking increase in terms of expression or in terms of number of the intestinal stem cell population in these tumors. So we have a lot of cells that are positive for LGR5 and also MAX3A, here identified by RNA in situ hybridization. And interestingly, we see loss of expression of the differentiation marker that I mentioned before, which is PPAR gamma. So at this point, we wanted to understand if this was a function, if there was a functional effect of MAX3A on this process. So we crossed these mice with our MAX3A knockout mice, and we can see that when we have a downregulation of MAX3A, the tumor burden or the tumor load in these mice is reduced when compared to the APC-KRAS model. Interestingly, at this point, we wanted to do functional assays to try to understand the relationship between MAX3A and also the PPAR gamma differentiation marker that I mentioned before. And so for that, we used tumoroids derived from these mice. I will call them AK and AKM for short, just to simplify. So the AK are the APC-KRAS mouse model, and the AKM, the APC-KRAS MAX3A knockout. And what we observed rapidly is that the culture is quite different in terms of morphology. So the AK tumoroids have this very cystic morphology, typical of a population that is more stem-like, while the AKM tumoroids, we have a reduction in the number of these cystic structures, and we start to have what we call budding tumoroids, which are, well, more closely resemble what we have in the normal mucosa. So we quantify this, and we could show that actually this tumoroid population has altered growth dynamics and morphology. But what is the impact of the PPAR gamma pathway in this phenotype? So to address this, we use the tumoroids for functional assays. So we treated the tumoroids with a PPAR gamma agonist called rosiglitazone from the glitazone family. And I will show you here the results of the expression analysis that we did for stem cell markers, here identified as LGR5 and LRE1, and using ANGPTL4, which is a PPAR gamma transcriptional target, as a readout of the treatment. So when we look at the AKM tumoroids, we already see that they have a slightly reduced expression of LGR5 and a trend for increased expression of the PPAR gamma signaling. When we treat the tumoroids with rosiglitazone, you can see that the AK tumoroids start to have a profile that is more similar to the AKM non-treated tumoroids. And when we treat these AKM tumoroids, the phenotype goes even further, and we see a striking reduction in the level of expression of these stem cell markers. So essentially, we could use the tumoroids here to prove that the absence of MAX3A directly impacts the expression or the activity of this signaling pathway. And so these tumoroids are more sensitive to PPAR gamma pathway activation. So in some way, this pathway is partially responsible for the phenotype that we observe in these mice. Obviously, this was done in mouse tissue, so we wanted to test this in patient-derived organoids. So we started a collaboration with IPO Porto to implement a patient-derived colorectal cancer tumoroid biobank. These are our current numbers. So we have a good number of tumoroid lines already expanded and cryopreserved. We have a success rate in the establishment of 68%, which is in line with what we find in the literature. And we started to use these patient-derived tumoroids to perform gene editing experiments with CRISPR-Cas9 technology to knock out the MAX3A in these tumoroids. We could establish knockout clones, as you can see here. Immediately, we saw that when we knocked out the gene, we started to have a higher level of expression of the PPAR gamma protein. This was we saw this by Western blot analysis, but also by immunohistochemistry. And accompanying this loss, accompanying this increase in PPAR gamma expression, we saw striking loss of the LGR5 population. And functionally, this has an impact because when we treat these tumoroids with the standard of care chemotherapy, which is FOLFOX, which is a combination of 5-FU and oxaliplatin, we do see that the MAX3A knockout tumoroids have a higher sensitivity to this treatment. So basically, with this part of the story, we then did other experiments that I cannot show you for the sake of time. But basically, we saw that MAX3A seems to play an important role, particularly in these early stages of colorectal carcinogenesis. We also analyzed its expression in about 200 colorectal cancer patients and saw that it was overexpressed in over 75% of the cases, with a negative correlation with PPAR gamma expression. And mechanistically, we could also determine that actually MAX3A binds directly the PPAR gamma transcript and regulates its signaling. And this has implications, as I showed you, for tumor cell morphology and behavior. So we think that this stem cell plasticity blockage, when combined with chemotherapy, might be an effective therapeutic strategy for colorectal cancer. So the other second part of the story that I want to show you real quickly has to do, as I told you before, with the molecular basis of chemotherapy resistance. So in colorectal cancer, like in many other cancers, there is a subset of patients, and here I'm using the stage 3 as an example, that despite being treated with surgery and adjuvant chemotherapy, they will still relapse. And this is quite a significant percentage. We are talking about 30 to 40% of these patients that will relapse in 2 to 3 years after initial treatment. And despite this type of chemotherapy being used for decades now, we still do not know what are the exact molecular drivers of this chemoresistance. There are some studies showing that RNA-binding proteins might influence the expression level of targets that are associated with this resistance. For instance, with the expression of drug efflux transporters. MicroRNAs might also be involved. So we decided to take advantage of our models and try to study the RNA binding proteome in this specific context. So for that, we analyzed the chemosensitivity of some of our tumoroid lines. So we could show that they have different chemosensitivities. So for the same drug within a single line and also for different drugs that we use. And we started to establish chemotherapy resistant tumoroids by exposing them to increasingly higher drug concentrations for long periods of time. So we were able to establish several lines that are either oxaliplatin resistant or 5-FU resistant. We can see that the IC50 increases in these cell lines range from 13-fold to 40-fold in relation to the parental lines. We also can see cross-resistance phenomena because oxaliplatin-resistant lines also show resistance to 5-FU and vice versa. And we have been characterizing these lines. One of the things that we did was bulk proteomics. So obviously we found differential expressed proteins in these models. And interestingly, we found a group of proteins that are common to all of them, which might indicate that they are really associated with the drug resistance phenotype. I cannot show you which exactly these proteins are, but because we are validating them, so this is still unpublished. But some of them do not have a described role in the literature, either in colorectal cancer or have not yet been associated with chemoresistance. So we are quite interested in studying these ones. What we also find interesting is that when we compare two of these oxaliplatin-resistant lines that have been kept for different time points in culture, we see a difference in the protein profile. So this OxyR35 cell line, it shows a protein enrichment for terms related with signal transduction metabolism and, interestingly, ribonucleoprotein complex biogenesis, and they show altered RBPs. And this profile is not observed for this OxyR55 cell line, which has been kept in culture for a longer period of time, which shows an enrichment for proteins associated essentially with DNA damage repair. So our preliminary analysis starts to show that maybe specific RBPs might be specifically related with this adaptation process to chemotherapy throughout time. And so we are using a technique called orthogonal organic phase separation, or OOPS for short, where we are trying to identify these specific RBPs associated with the drug resistance phenotype. This essentially, we perform UV cross-link to stabilize the protein RNA interactions, and then we perform several rounds of phenol-based phase extraction to really get RNA-RBPs complexes, which are then isolated by RNase treatment. And we have optimized this technique using our resistant tumoroid lines. And so now we will perform proteomics also on these models to try to pinpoint the exact RBPs that are changed in these resistant lines. So to conclude, we have generated for the first time actually these drug-resistant colorectal cancer patient-derived tumor lines to study these resistance mechanisms. We think that this RNA-based control of gene expression might be strategically advantage to cancer cells, which are facing adaptation to chemotherapy. And this is important because we know that these processes are reversible up to a certain point, which might provide a therapeutic opportunity. And we are also trying to now start some projects where we are trying to use these models to assess the treatment response to drugs or drug combinations before they are actually administered to the patients. And in the colorectal cancer context, this is possible because our turnaround time to establish these models, expand them, and test, for example, their response to drugs takes about 8 weeks, which is perfectly in line with the time to make a medical decision regarding, for instance, the application of adjuvant chemotherapy. So we think we can use it to try to see if you can predict the clinical therapy response. So these were the two stories that I wanted to show you. And finally, just the final slide with the acknowledgements. The first part of this work was mainly performed by Rita Silva, which is a PhD student, and the second part is being led by Alexandre Coelho, another PhD student, with contributions from Morgan and Vanessa. Also thanking all the platforms at I3S that have been helping us, our collaborators at IPO, the patients, our external collaborators, and obviously the funding agencies for supporting us. So thank you.
Thank you so much for the nice presentation and sharing all of your results. Does anyone have a question for Bruno?
Hi, hi Bruno.
Hi, Sarah.
Very nice presentation. I really liked it. I'm not very familiar with cancer or this type of research, but I hope I can ask an interesting question. I was very curious about your last part where you created these drug-resistant tumoroids.
Mm-hmm.
And I was wondering, because I guess, how do these compare to real patient-derived organoids who have relapse after the treatment, or what are the differences that could be between these?
Sure, that's a really good question. So most of our tumoroid lines that we have were derived from treatment-naive patients. So we designed the protocol with IPO from the start like that because we already had in mind to establish these type of models, and we didn't want to have confounding factors from previous treatment. But we have 2 or 3 lines that actually came from patients that received neoadjuvant therapy. So they were first treated with chemotherapy, and only afterwards they had surgery to remove the primary tumor. And our preliminary comparison, when we compare those models with the ones that we have generated, is that the ones that are derived from patients that have been treated already show a higher higher baseline resistance to the drugs compared to the lines that have never been exposed to the drug before. So we have not gone very much more than that point. So we don't know if for example, the protein profile, the protein expression profile is similar or not, but this is something that we can certainly compare in the future to make the point that the protocol that we are using to generate the resistant lines somehow can mimic what happens in at least in patients that have been treated. Comparing it to patients that relapse will be more difficult because it is difficult to have access to those samples, mostly because when they relapse, you already have metastasis or advanced stage, and it's harder to get those samples to establish the organoid lines. But we will try to do it if possible, because that would be the best model to compare with the ones that we are generating. Yeah.
Okay, because once they relapse, they are refractory to treatment, right? Or
yes, some of them will be, others won't. But at least for the broad chemotherapy drugs that we use, certainly they will become more resistant, and in many instances you have to go to targeted therapies. So it will depend on the mutational profile of the tumor after the relapse. Yeah.
And more curiosity, since this is a bit more my field. What do you know, or is there any knowledge in vivo or in patients about the effects of gut microbiome on this?
Yeah, so that's an expanding field in the organoids. So you start to see an increasingly high number of studies employing these organoids to study the effect of the gut microbiota. not only the normal flora, but also the effect of pathogenic bacteria. So they are, most cases, they are amenable to infection. You can study the infection process. For instance, we have a collaboration with another group in I3S, the group of Didier Caban, that is studying Listeria monocytogenesis infection. And we use these organoids, we are now using these organoids to infect with Listeria and study the dynamics of the infection process. And you start to have more studies that are trying to make these type of co-cultures, complex co-cultures, some of them also using, trying to mimic the complex microbiota that we have in our gut, to try to see how the different species compete among themselves and what is the impact in terms of the gut homeostasis. So yeah, it's being used, yeah.
Okay, when you come for the interim, we can talk more.
Sure.
Thank you.
Thank you.
More questions?
Hi, I'm Joanna.
Hi.
More of a curiosity because I also don't know anything about cancer. So there might be silly questions, but so you see this more as a predictive tool for relapsing. So knowing if this patient is more likely to relapse and then aiding the therapeutic, so following them up closely, or if it's more as you're making the decision on the clinics, deciding for the best therapeutical option. And if that's it, how do you think it compares to this zebrafish avatars that
Sure.
are being produced in Champalimaud Foundation?
Yeah. So great question. So actually they are being used for both instances. So you already have some publications showing not only in the colorectal cancer context, but also for other cancer types, that you can use these tumoroids to really test what will be the clinical response of the patient. For some of these studies, the level of correlation in terms of the outcome that you get from the tumoroids and the patient response is quite high. So we are talking about 70 to 80% correlation in some studies. So the indications are that they might be promising to be used in that context. There's not so much concerning the relapse effect. So that came to us because we wanted to study resistance. So we are trying to see if it can also be used in that context. How it compares with the zebrafish. So I think they might be used in a complementary fashion, actually. So as far as I know, the zebrafish avatars, they allow you to do a quick, relatively quick screening of the effect of the drugs on the cells. And I think the tumoroid models in this case might be more useful if you want to try to do this type of more molecular biology approaches where you need a lot of cells to do proteomics or something like that. But in terms of assessing in vivo effect, I think the zebrafish is great. It might be better than using, well, at least it's cheaper than using mouse models, I guess. So,
yeah, I think for the quick therapeutic decision, yes, but I think your model then has the advantage of, as we were talking,
yeah,
microbiome, also maybe co-culturing neurosystem. What is the role there, and then having more predictive factors.
Yeah,
but
and
so
also,
like
yeah,
using
as
both.
you mentioned, yeah, and also as you mentioned, it's being widely used for drug screening approaches. So to try to find in cases where you really don't know how to treat, or there is no or the patient is refractory to the drug that is usually used in standard of care treatment, to try to find alternative drugs or repurposed drugs. So there's the field is quite active also on that front. Yeah.
Yeah, very nice presentation. Thank you.
Thank you.
Okay, thank you so much. We
Thank you.
move on to Sandra Tenreiro presentation. And okay. So Sandra Tenreiro holds a PhD in biotechnology from Instituto Superior Técnico and is currently the group leader of the Generation and Aging Lab at NMS UNL. She is also the NMS coordinator of the MPS Nova Training Project. Her research is focused on clarifying the molecular mechanism of neurodegeneration in the context of aging disease, such as retinal degeneration, using in vitro models such as retinal organoids. She is particularly motivated to explore alternative models to traditional animal research methods. She is also collaborating with ophthalmologists and actively involved in clinical studies. Sandra has one patent under review and published more than 60 publications in peer-reviewed journals. And today she is going to talk about organoid-based new approach methodologies for early intervention in retinal disease. Thank you, Sandra, and the stage is yours.
Thank you. Thank you, Juliana. Okay, I'm going to share my screen. And are you hearing me well?
Yes.
Yes? Okay. Are you seeing my screen already?
Yes.
Okay, great. Okay, thank you for the opportunity. It's a pleasure. Of course, I'm very motivated to share my research here at this very interesting twinning project that Sarella put running. Okay, so I'm going to show you some of the research we are developing, more specific, using organoids to understand diabetic retinopathy. And oops, okay. Okay, so starting to introduce you to diabetic retinopathy. So this is a common complication of diabetes that is, as you know, a public health challenge of our century. And it's the main cause of blindness in the working age population. So it's really a very impactful disease. Just to give example, our vision is affected in this black dots in the visual field that starts to interfere. So these patients are not able to see faces, for instance. Unfortunately, the therapeutic options that are available, they come too late. They are targeting only the latest phase of the disease, where there is neovascularization. And also they are invasive, as you can see, and they are not able to restore vision. So really, this is the big limitation in the field. So what we need are interventions for the early stage of the disease, where we still can change the course of the disease and avoid blindness or vision loss. So here is a diagram just to show you how this disease evolves. This is a disease that can take 10, 15 years to evolve, depending on the metabolic control of the patient. But unfortunately, 1/3 of the patients after 5 years of diabetes diagnosis, and we must have in mind that before these diagnoses occur, there is a long period of pre-diabetes also. So 5 years after the diagnosis of diabetes, 1/3 of the patients already have alterations in the retina. So the retina is this thin tissue in the back of the eye, and it's where we see these alterations. And in the first stage, where there are no clinical manifestations, we know now that there is already inflammation, gliosis, neuronal dysfunction. And after this period, we have what we call non-proliferative diabetic retinopathy, where we start to see alterations in the vascular system. In particular, there is cell death of the endothelial cells that lately will result in loss of functional capillaries. And in the later stage, the proliferative diabetic retinopathy, we have neovascularization and edema. And that's only for this stage that we have some therapeutic approaches that are really just to avoid the progression or that the disease become even worse. Usually they are based on anti-VEGF, vascular endothelial growth factor therapies, and they are invasive because patients need to have intravitreal injections once a month or even or each 2 months. So what are the limitations in the field and why we don't have therapies for the early stages? Mainly because limitations related with the kind of models we have available. For many years, researchers were using and are using and still use rodents models. But as you can appreciate, mice retinas are different, and the eyes also are different from the human eyes. One thing that is important is that mouse are nocturnal animals. So the photoreceptors and also the circuits and the specific cells in the retina are different from the human specific retinal cells. And also in the human retina, we have a specific region that is really important, that is the macula, and where it is fovea, that is absent in the rodents. So for this reason and others that are the lack of translation between the models we have today, not only in vision, but in general as well, we know that 80% of the drugs that reach clinical trials, they are going to fail, or they are failing so far, because 60 of these drugs, they fail because they lack efficacy that was evaluated in preclinical studies, in preclinical models, and the other 30% due to toxicity that was not identified in preclinical models. So that's why it's so important to complement these models, or to improve these models, or find new solutions to explore and to understand the disease, but also to find therapeutic interventions for the early stages. So in my lab, we are using retinal organoids. Very briefly, we obtain these organoids from iPS cells. We use a protocol that is a recent protocol from 2019. So just to mention, these retinal organoids protocols are still evolving. All months we have new publications showing new ways to improve these models. But the protocol we are using in the lab is based in this Kapovsky paper. We start by forcing ectoderm and neuronal differentiation. We obtain these embryonic bodies, and then we plate these embryonic bodies and in a matrix gel coated plate. And at day 30, we used to dissect these neurovesicles, but now we are doing a different way. We are just cutting and then selecting the beginning of. At day 7, you can see that we already have free-floating organoids that show some stratification. You can see that there is this differences in this image that can show that we have a beginning of a stratification. At day 180, if we wait enough, we can see here the facing out the outer segments of the photoreceptors. To be more exact and show you what we am I speaking about, this is the stratification that the human retina has. So we have different cells from the inner to the outer part of the retina. We have a very beautiful stratification and different cells organized in a very specific way. So facing out, we have the photoreceptors in close connection with this epithelium, that is retinal pigmented epithelium cells. It's very important to maintain the variability of the photoreceptors. We have then these photoreceptors, they do synapses with bipolar cells and two ganglion cells, and the ganglion cells are the ones that then through the optic nerve will deliver the information to visual cortex where the image is formed. In the middle, we have other kind of cells like horizontal cells, bipolar cells, and macrine cells. And we also have microglia and macroglia. Here we have a specific kind of macroglia from the retina, the Muller cells, that are really important. In the retinal organoids, we are able to reproduce all these different kinds of retinal cells, but we have limitations that I'm going to explore in a couple of slides, that mainly the RPE, the retinal pigmented epithelium cells, they are not organized around the photoreceptors that are facing out. They form these spheroids apart. This is a limitation that so far lots of labs are trying to surpass, but so far was not possible to have a really well-organized RP layer around the photoreceptors. Okay, so advantage of using retinal organoids. We are speaking about human models, and these relevant human models are predicted to de-risk the translation efficacy from preclinical to clinical in research and development, about 26% in the costs that are involved. We have stratification, as already mentioned, and here in this image it's clear that we have outer nuclear layer, an inner nuclear layer, and a layer of a plexiform layer with a synapse in the middle. In this iosine amoxicillin staining. We have stratification, we have all the neuroretin and glial macroglia represented. However, we have limitations as we have in all models. We have complex protocols. As I told you, we can wait 120 days to have our organoids to do our experiments. And we have limitations in heterogeneity and reproducibility. If sometimes even in the same batch of organoids, we have more or less stratified organoids, but also from differentiation to differentiation. Sometimes we have a necrotic core. It's not the case of this beautiful organoid here, because now we are doing an approach. We are processing these organoids to have them smaller, not allowing them to grow too much. We don't have microglia and we don't have vascular system as brain organoids. They don't have these because both microglia and vascular system is differentiated from the mesoderm, and because as I already mentioned, very soon we force differentiation to ectoderm, so we don't have these two systems, and again we don't have RPE around the photoreceptors. So we thought, okay, we have these organoids, they are really nice. We are very interested in diabetic retinopathy in the early stage. And maybe it's an advantage with the fact that we don't have the vascular system, because it's difficult to study these two different effects, the neurodegeneration or neurodysfunction, gliosis by one side, and the vascular system alterations in the other side, and how these two systems cross talk. How it's possible if we interfere and we do neuroprotection and avoid or reduce inflammation, can we avoid the alterations in the vascular system that we see later on? How these two pathways are interconnected. But so this was the first idea, and Luisa Lemos, a former postdoc in the lab, an amazing postdoc, was the person that put this model working. So she obtained organoids within 100 days. She exposed to higher concentrations of glucose to mimic hyperglycemic conditions. She had done a lot of experiments to reach to these concentrations and conditions, and she observed that at these precise concentrations, she was able to see neurodegeneration, here evaluated by two different readouts, epignotic nuclei and staining with fluorourate C that is marking neurodegenerating neurons. And she used as a control mannitol that induces osmotic stress that can be also expected that high concentrations of glucose can induce. But as you can see, this same equimolar concentration of mannitol is not doing the same effect of neurodegenerating, inducing neurodegeneration as the higher concentration of glucose that was here used. So she then evaluated different markers to evaluate the specific populations on the retina that could be neurodegenerating, and indeed she found that the macroin cells and the ganglion cells were affected with these higher concentrations of glucose. Photoreceptors, at least in the conditions we tested, didn't show to be affected by glucose, but I think that this is expected result because photoreceptors are consuming a lot of glucose, but maybe they are happy. I don't know. Maybe, also, we didn't test for longer periods of exposition. Also, we observed markers of gliosis. This means that the Muller glia is being activated, and we have here vimentin, a marker of Muller glia that we can see that is increased, and these processes are more pronounced. But also glutamine synthetase is increased. This is an enzyme that converts glutamate in glutamine that is released to the media, and here we also see that this glutamine is being released, showing overall these data show that we have gliosis. Also, we evaluate some chemokines and interleukines, and we observed that, for instance, MCP1 is also being secreted to the media, and EL1 beta expression is increased. And also we saw a very nice result. So if 1-alpha is the transition factor that is associated with hypoxia, here we don't see a significant increase in this expression level, but we see an increased level of VEGF. And this was an interesting result because in our organoids, we avoid to have RP cells that are one of the sources of VEGF. Another source could be the endothelial cells that we also don't have in our organoids. So in our organoids, we just have one kind of cells where this can come, that is the Muller glia. It means that after 6 days of exposition with glucose, Muller glia is already expressing higher levels of VEGF, indicating that at this early time point, VEGF production is already being induced, and this can be the beginning of a crosstalk with the endothelial system or the endothelial cells. So now what we are doing with this, we are doing different approaches, but one of that I would like to tell you is the one that Beatriz Felgueiras is exploring, that is to use this model to identify neuroprotector molecules or anti-inflammatory molecules that we can use at this early stage. She is co-supervised by Cláudia Nunes dos Santos from NIMSB, and she's working with human phenolic metabolites that Cláudia identified in her models in microglia, 2D cultures, but also in animal models that these compounds can be neuroprotective. In particular, in this case, it's a compound that is more anti-inflammatory. And so again, using vimentin as a readout, we can see that glucose increases vimentin levels or Muller glia, gliosis, that is relieved with the treatment with these two different concentrations of this compound. And here are the quantifications for the intensity of vimentin and also the evaluation of the nuclei that show condensated chromatin. You can very easily see that there are here a huge dispersion in our data because we didn't want to remove anything, but these two are two different differentiations, and as you can see, we are facing this problem that is the variability between differentiations. Are you hearing me, or I am frozen?
No,
You are
it's okay.
good.
Sorry, it's okay?
Yeah.
Yes.
Okay. Okay. And here in the cell death, it's interesting that we see for the higher concentration and protection, or at least, I think if we do this treatment, at least for not only in the latest 24 hours, but maybe 48 hours, maybe we have a stronger effect in neuroprotection. Anyway, maybe this neuroprotection is an indirect effect because we are relieving inflammation. So we are now completing this data with the ELISAs and so on. Maybe this is just an indirect effect. So we are now, Beatrice is exploring this data. Another thing that Beatrice is working, and this is in collaboration with the Max Delbruck, with Agnieszka, is to improve or to increase the complexity of our model. The idea is to add microglia and the vascular unity to our retinal organoids, and in parallel to add the RPE and to do a kind of culture between our advanced model with microglia, vascular system, and RPE, and then work and improve this diabetic microenvironment, not only exploring higher levels of glucose, but also insulin that is increased in prediabetes, and by adding to the media fatty acids that are known to be increased in the circulation of diabetic patients. This is the project from Luana, and these two projects are being funded by MPS Nova under the exploratory projects working package. I think this is going to be very, very exciting. For now, I just have data from this part regarding RPE. So what Beatrice have done, she took these RPE spheroids, she dissociated these RPE spheroids and put them in a monoculture and followed this monoculture for 60 days. Are you hearing me? Because I'm frozen in my computer.
We hear you, yes.
Okay,
Yes.
if you have problems, just let me know. So,
Okay.
when we compare this culture with the cultures that usually we have in the lab, that we use in different projects, from the most common RPE cell line, the RP19, or primary porcine RPE cells, or even RPE that is directly differentiated from iPS cells, or these ones that we normally don't use just for specific required experiments that are primary human fetal RPE obtained from Lonza. So you can appreciate that the RPE differentiated from organoids that Beatrice obtained are more pigmented, have a more icosahedric form. They are more similar to adult porcine RPE cells. This is promising because means that we, in these RPE that are maintaining culture with retinal organoids, maybe it's known that trophic factors can be here helping the maturation of both the retina and the RPE cells. So she just have done some validation that these RPEs are expressing markers of that are known to be specific for RPE cells, and also that they are phagocyting outer segments of the photoreceptors, because this is the function these cells do in our retina. And indeed, here you can see these autofluorescent granules that are coming from the outer segments phagocytosis. So we feed these cells with outer segments from porcine eyes. And also these monolayers behave as a barrier because they show increased level, increased tear, so trans-electrical epithelial resistance over time. So what we want to do with this also is to develop a platform for therapeutics discovery and development. So we want to optimize our readouts, to avoid manual handling, and to do to explore these readouts of this early diabetic retinopathy model, to convert them in, for instance, more automated flow cytometry readouts, or even multiplex readouts, to be able to have here a pipeline for drug screening, and to offer these as a collaborative or contract research. But also we want to enlarge our proof of concept testing, and we want to do this also integrated in the new preclinical hub we are developing here at Nova Medical School. So this project is submitted to La Caixa. Past week I went to the second phase interview. Let's see if we have funding to implement this pipeline here at NMS. So as a summary, I would like to reinforce that the main ideas are that the retinal organoids mirror the human retina, are human-based, supporting preclinical development and higher translational accuracy, and that they are more ethical and cost-effective, and a good alternative to animal models, alternative of complement, because I think some questions will need anyway to use animal models, but we can drastically reduce the animal experimentation by using more retinal organoids. Our model of diabetic retinopathy mimics the key features of the early stage of the human disease, and we can use that, and as I'm showing you here, we are exploring that in different ways, also in the context of the other twinning project we have at NMS, the EFCA, and also to dissect the disease mechanisms that are not so explored. I would like to take the opportunity also to tell you about this advanced training that is being offered also to the MPS participants. So this is an advanced course in different kind of organoids and microphysiological systems. We are very happy to count with Agnieszka as well in this course that will take place in November and will be completely online, so everybody can join. And also I would like to take the opportunity to call your attention to the new cost action that I'm very happy to lead, that was recently funded, and we count already with more than 100 members, and more about 30 European countries and Canada, United States and India. We count also with patients associations and SMEs, and everybody can join, if everybody interested in retina, by going to the cost action page in the from the community, European community, and apply here. So it's very easy. I would like to thank my group, my great group, Beatriz in particular, and Luana, that were the persons involved in the work that I show you today, but also to my collaborators, in particular Agnieszka and the other collaborators that are giving me all support to follow with my research. Please let me know if you have questions, and thank you for the opportunity.
Thank you so much, Sandra. Very interesting work. Does anyone have a question for Sandra?
I have a few questions, but maybe for the sake of time, I can also ask you later, because I know Sandra, so maybe let's give priority to someone else.
Okay, Bruno.
So Sandra, really nice work. Congratulations and good luck for the La Caixa application. So this is just a curiosity. So your RP monolayers, do you have the retinal organoids floating in the same well? I didn't quite caught that part.
Yeah. So before we do, yes, we have when we have the spheroids of RPE, they develop together with the retinal organoids, and usually they are really attached to the neuroretina, but they form like a spheroid in the side of the organoid. What we do, we cut them to do our experiments, to don't have the RPE there. For some experiments we do it, but we maintain them in the culture because we have the experience that if we have RPE there, it will be better also for the neuroretina development and maturation. When we want to do a monolayer, as I show you, what we do, we take the spheroid, we dissociate, and we put it in a monolayer. We can do it in a transwell, for instance. And then, if we want to add the organoid on top, then we will need to do some kind of a matrix to force the organoid to stay in contact with the RPE. We already tried to do that because it was published by Kevin Ashberger from Tübingen, but it's not so easy to do as in the paper because then you have. So when we try then to fix, to prepare, for instance, to do slices for immunofluorescence, they go apart. It's very easy to go apart the photoreceptors to detach from the RPE. It's exactly what we have when we use animal highs. The retina slices, it's a very fragile structure, and the RPE detaches very easily from the photoreceptors. So we have the same in the organoids. There are several groups that are trying to do a U-bottom cover with RPE and then the organoid on top. There are different designs, but so far there is no robust protocol to do that.
Or using transwells maybe, or have people tried it?
Yeah, they have done in transwells, yes.
Okay.
But
it's difficult, yeah.
No, it's not, yeah.
Really nice.
Thank you.
Thank you so much, Sandra. There is any more questions?
Joanna, just one question from you. It's good to have you here.
I have so many. Okay.
I'm
going to the one
like
that I think is more interesting, and then the others we can discuss later. So in your high glucose model, I do think that photoreceptors love glucose, so they will not die. But what I think that you will maybe have, and I don't know if you are looking for, is you are losing layering. They are not so nicely layered, so your cells are being displaced. So you can clearly see your ganglion cells are no longer on the center.
I'm not hearing you.
Can you hear me now?
Hearing you very well. Can you repeat?
Yeah.
I'm seeing in my screen is everybody frozen. So can you repeat?
Yeah, sure. Can you hear me now?
Yes.
Yeah. So you mentioned that you don't see a difference in your photoreceptors in your organoids because they probably like glucose. True. I've cultured my photoreceptors with higher glucose than you do, not in the disease model, but as in the control, and they love it. They are super active. But I think the fact that we don't see loss of photoreceptors doesn't mean that there's no reaction, because you lose layering. Your structure is definitely not as nice.
Yes.
And you see a lot of displaced cells. So you even had ganglion cells at the edge, which is crazy.
Yes.
So it's interesting.
So
it's something that is.
So one thing is cell death. Another thing is dysfunction.
Yeah.
Dysfunction. Cell death is the ultimate phenotype. Dysfunction is something that starts to occur much earlier. And here I'm not evaluating dysfunction. We were looking for cell death. Okay, cell loss
Cell
with the
death.
marker.
Yeah.
So, yeah, and so you are right. You are completely right. Doesn't mean that we don't have dysfunction, and I trust that even we have dysfunction. However, it's more harder to evaluate. It will be great to have systems like microphysiological MIA system working
MEA.
to do electrophysiology, to use other markers of stress. But we tried for case-based tree, for instance, is not showing a robust result, for instance. So we need to go to other kind of readouts to evaluate dysfunction, because indeed dysfunction is what occurs first. Yeah, you are right.
Yeah, I think it's worth to also look at the maturity markers, because maybe they were there and then photoreceptors, they are happy, but they downregulated. They are just like, I'm not going to produce this right now.
Exactly.
And that's
at
why
this
they
stage,
are.
I'm saying, yeah, and at this stage, 120 and 110, what we have is photoreceptors progenitors. So it's not really photoreceptors, but yeah, you are completely right.
Would be very interesting. Okay. The other questions I wrote down, I will email you.
Okay, thank you. Great.
Thank you. It was a great presentation.
Okay, thank you so much, Sandra and Bruno, for excellent talks, and thank you so much, everyone, for assisting. And I hope to see you all in our next virtual meeting on 18th of September. Thank you so much. Have a nice day.
Thank you.
Thank you.
Thank you.
Bye-bye.
Bye-bye.
Thank you.
Bye-bye.
Thank you, Bruno.
Bye. Bye.
Thank you.